Elita Jauneikaite, Assistant Professor in Microbiome and Bacterial Genomics at Imperial College London in the UK. Today I will share with you some of the work that we're doing with colleagues at Imperial College London and UK Health Security Agency to investigate the mobile genetic elements driving carbapenemase genes in the Enterobacterialis in the UK. Carbapenems are the broad-spectrum beta-lactams and usually are used as a last resort to treat multi-drug resistant infections. Carbapenemase-producing Enterobacterialis infections are increasing globally, and in particular, the rates are increasing in the last few years. Primarily, carbapenemase genes are VIM, KPC, NDM, as well as IMP. In our previous regional study, we have investigated an outbreak within the regional network of hospitals that was driven by IncHI2 plasmid carrying IMP1 and MCR9 genes. So to answer our question, we have analyzed 208 high-quality whole genome sequences of carbapenemase-producing Enterobacterialis confirmed to be carrying IMP gene from the UK between 2014 and 2022. We used an extensive bioinformatics analysis and various bioinformatics tools to characterize our genomes. Within our analysis, we have identified in total of 18 bacterial species within the Enterobacterialis, with 3 of them being the dominating our data set. It's Enterobacter hormacii, Klebsiella pneumoniae, and Escherichia coli. So altogether, these species accounted for 81% of the species identified in our data set. In total, we have identified 6 alleles of blaIMP gene, with IMP4 and IMP1 being the dominant in the data set. Reconstruction of the plasmids present within the data set has showed that primarily we had plasmid IncHI2 present in our data set, carrying blaIMP4 allele, as well as blaIMP1 being the second most dominant allele identified on these plasmids. 89% of IncHI2 plasmids carried blaIMP as well as MCR9. Further analysis also identified that there were heavy metal resistant genes present on the plasmids as well. And even though we identified a couple of lineages of IncHI2 plasmids present with our data set, primarily they had the same genes present on the plasmid. At the moment, we have confirmed that IncHI2 was the main driver of blaIMP genes within the Enterobacterialis dataset that we have analyzed, and we are doing further analysis into further characterization of these particular plasmids. I also would like to thank all of the colleagues who have contributed to this work, and in particular on behalf of myself and my colleagues working on this project, we would like to say thank you to all the hospitals that are sending isolates to UKHSA that allow us to do this research and understand the spread of antimicrobial resistance within the healthcare-associated bacterial pathogens.